Well, good afternoon and welcome. I'm Chris Fanta. It's my pleasure to welcome you to our asthma grand rounds this afternoon. We have really a very exciting topic and a panel of speakers, and I'm pleased to introduce as a moderator for this program, Dr. Scott Weiss, who, as you know, is sort of revered mentor to many of us in the audience today. He is Director of Respiratory, Environmental, and Genetic Epidemiology in the Channing Division of Network Medicine. He's Director of Partners Center for Personalized Genetic Medicine and a professor at Harvard Medical School and Harvard School of Public Health. And we're delighted to have you introduce the program and moderate it for us. Dr. Scott Weiss.
Okay, welcome everybody, and thank you so much, Chris. So the topic today is in utero influences on the development of asthma, and we've got three speakers, Kathy Haley, Sunita Sharma, and Kelan Tantisira, who are going to be talking for about 12 minutes each on various aspects of this topic. And I just wanted to have a couple slides to introduce this. So as background, this is work that was done for an R01 grant, Intrauterine Smoke Exposure and Asthma Genomic Origins, that Les Kosbik and I were the PIs for, and the goal of this grant was to integrate immunologic and mouse models with human transcriptomics of fetal lung development. So it was an integration of human transcriptome with mouse transcriptome and mouse models for asthma. We have a population of over 500 human fetal lung samples with gene expression data, GWAS data, and methylation data, and Kelan and Sunita are going to be talking about that, and then we applied this in the mouse to look at the molecular mechanisms. So in terms of background, regulatory variation in the genome is strongly associated with complex traits. That's one of the lessons from GWAS. And many genes that have been discovered are involved with fetal lung development. And exposures that influence methylation of fetal genes change fetal programming and potentially then the course of health and disease for that fetus going forward. And there are a number of examples of this. In utero cigarette smoke exposure, which is the example that the grant was based on, but also vitamin D, folate, B vitamins, vitamin C, retinoic acid, and omega-3 fatty acids. And the reason vitamin D and vitamin C are in bold on this slide is that there are clinical trials underway or published that relate the use of these vitamins in utero to modification of outcomes in the fetus and modifications of asthma. So our three speakers are each going to speak for about 12 or 13 minutes, and then hopefully we'll have time for questions, panel discussion at the end. So Kelan is going to kick it off and just talk about transcriptomics of fetal lung development.
All right, thanks. I appreciate the invitation to speak with you today related to genomic factors influencing normal lung development, and specifically for this audience, their relevance to asthma. This is just a brief outline of the points I'd like to cover today. Certainly talk a little bit about normal developmental lung transcriptomics, and then look a little bit at the association of the genes expressed during the stage of human branching morphogenesis, a time of airways development, and obviously the thought process being that they may be more likely to be related to asthma from that perspective. And also talk a little bit about developmental pathway enrichment analysis as it applies to vitamin D, the vitamin D pathway and the corticosteroid pathway, both pathways which have been implicated certainly in the asthma susceptibility as well as in overall lung development, and end up with a couple of concluding statements. For those of you who are not statistically inclined, I wanted to put up one slide describing principal components analysis, because a lot of what we've done has focused really on principal components in terms of their ability to determine the maximum variance of data, and by allowing aligning your data such that genes determine the maximum variability gives you a lot more information in terms of which genes have the highest probability of being related to a given disease state. So this slide may be familiar to you, but basically the developing lung proceeds in 4 primary phases: pseudoglandular, canalicular, saccular, and alveolar phases. For our particular project, the human interrogation of those samples really is limited to the pseudoglandular and maybe up until mid-canalicular, because we can only obtain aborted fetuses up until 20 weeks of gestational age. Having said that, the nice thing about our approach is that we can also refer to the murine model for more complete development throughout all of the developmental stages, and Kathy will certainly be lending some insights towards that. So our initial analysis, which was published a few years ago now, was more of a proof of concept, and we were asking, what can the molecular development of these early lung samples really tell us in terms of what we know histologically and what we may not know genomically. And so in this study of 54 samples run on some Affymetrix gene expression arrays, the big thing that you can see, 2 take-home points. One is principal component number 1, which is the x-axis. You can see that that actually does represent developmental time. Samples, the numbers represent the age of the samples, estimated gestational age in days. Clearly, all of the younger samples are on the left, all of the older samples are on the right. But in between there, I'll point you to a number of these areas where you see sharp changes in the overall transcriptomic profile. The interesting thing that we found initially is we initially focused on the 117-day transition because for whatever reason that corresponds precisely to the traditional cutoff between the pseudoglandular and the canalicular stage of development. And to us, that was kind of a nice proof of principle that something's going on there, that histologically there are certainly changes, but molecularly there are certainly changes as well. Now, we also noticed, similarly, that there was this nice transition point around day 94 that we weren't sure exactly what it was, but clearly there were a lot of molecular changes going on, a lot of changes in gene expression, and initially we thought that it might be related to some sort of gene priming, getting ready for transitional stage transition. And as you'll see later on, it may also be related to some other things as well. So Sunita went on to look at some of these early developmental SNPs and just to see if they were associated with lung function, given the prominence of airways development during this time. And I'm not going to go through this slide in any detail, but you can see that the genes that are most tightly correlated with lung development are also, a lot of them are significantly associated with airways phenotypes in multiple independent asthma populations. So, more recently, we have, and this is unpublished so far, the manuscript is in preparation, updated this analysis to include 139 samples, a little bit younger on the age of the fetal spectrum, and we actually focused this specifically to say, well, maybe there are roles independently to male and female development as well. I'll point out 2 things to you. One is we still see this nice molecular transition at around day 94. We are not powered to see the second transition at day 117 because we actually had very, very few samples that were older than day 117 in this particular analysis. The other thing that I'll point to you is that, again, gestational age is a large part of the principal component. But when you put males and females beside each other, the males are in the magenta or purplish colored samples, and the females are green, you can clearly see that there are differences in males versus females in terms of the overall dominant gene vectors in principal component number 1. So, put this another way, when we actually look at the overall contribution to principal components, sex is actually now the single largest determinant of principal component number 1 and number 3, whereas gestational age takes up a lot of what we would call principal component number 2. So they're both tightly contributing, both sex and gestational age to overall airways development. The slide on the right, the picture on the right, just basically says, well, what happens? You know, at a given age, does it look like the males are contributing more to the gestational age in terms of more maturity than females? And those are represented when the slides are pink, and or is it females that actually are contributing to the dominant variation at a particular gestational age? And the interesting thing here is we kept on talking about this molecular transition at day 94. And here, in fact, this day 94 to day 96 pops up very prominently here, suggesting that that potential molecular mechanism may actually be the fact that genders are actually starting to influence fetal lung development from a genomic perspective, and that could actually be a gender transition stage as well as a molecular transition stage. So if we actually take a look at this updated set of genes, and related to asthma gene expression, and this is just a publicly available asthma gene expression set, you can see that principal component number 2, which is again gestational age, is certainly modestly associated with an enrichment in asthma genes. So as the genes that contribute to branching morphogenesis, again, related to asthma susceptibility. Principal component number 3, which is predominantly gender-based, is also, and if anything, a little bit more enriched for asthma susceptibility overall in terms of the proportion of genes that are both developmental genes, developmental sex-related genes, and related to asthma. Okay. So in summary, for this initial portion, normal molecular development, certainly principal component analysis, has revealed consistently this novel molecular developmental stage. And during early lung development, sex and developmental age are primary drivers behind this molecular signature. And these genes that are associated with branching morphogenesis have consistently been shown to be enriched in terms of asthma susceptibility genes as well. Okay? So for the second half of my talk, I wanted to talk about fetal developmental pathways in asthma and give kind of a methodologic overview as to how we approach this. First, you identify a pathway of interest. In this case, we'll talk about both vitamin D and corticosteroid pathways. You can identify significant genes within the pathway themselves, and then map those genes to say, are they developmentally regulated, and identify the subset of those genes that are developmentally regulated. And finally say, okay, well, you have a set of developmentally regulated pathway genes. Are these enriched or not enriched for asthma susceptibility? So, there have been a number of prenatal cohort studies now looking at the influence of vitamin D. This is only one of them, but they all show the same thing, that basically lower maternal vitamin D level is associated with significantly higher levels of subsequent childhood wheezing and or asthma. Okay? And here you can see that the odds ratio higher versus lower is about 2.5-fold higher for those within the lowest vitamin D versus the highest vitamin D. So what we did is for vitamin D, we took 2 data sets. One is we just said, okay, let's identify all of the vitamin D related genes by what we know in terms of terminology. And so we did an ontology analysis looking for cholecalciferol and everything related to it. And then we also took an unsupervised approach where we used an independent ChIP-seq data set to look for vitamin D regulated genes. And we put them all together here in terms of supervised versus unsupervised lists. We plotted those out subsequently in principal component space, and here they are on the right. And you can see that there are a variety of genes that are at the extreme polar left and polar right of the principal component space that would be of interest to us in terms of overall inclusion within our analysis for highly developmentally regulated vitamin D genes. So from there, we looked in both the murine model as well as the human model, just basically to identify the genes that are in each principal component, in the top 5% of each principal component, and then look for a common set of developmental genes between the mouse and the human. And we identified 38 genes that were common between the murine and mouse developmental data sets that were vitamin D genes. You can see that these genes were developed, and the heat map will show that they're differentially regulated across time. It's kind of nice, and a little bit misleading because human data sets, these are all early developmental stages, and the murine data sets include the postnatal ages as well as the early ages. But having said that, taking these 38 vitamin D regulatory genes And when we looked at it with in relation to asthma, we see that they are significantly enriched with an odds ratio 3-fold higher than what you would expect. And so, again, vitamin D regulatory genes certainly look like they have an influence overall in asthma susceptibility. Last topic we'll talk about is just corticosteroids and their overall need for normal lung development. Now, this is just a corticosteroid, glucocorticoid positive versus negative developmental mouse, and you can see that steroids clearly are needed for normal human lung development. And we took a very similar approach to what we said previously, except in this case, well, we took more of a union approach. We took 3 independent data sets related to differential expression by glucocorticoids. One was a ChIP-seq experimental data set. One was derived from 164 lymphoblastoid cell lines from asthmatics. And then one was a murine lung development data sets. And we thought that we would define the significant glucocorticoid genes as any genes within 2 of the 3 data sets. We also plotted these out in principal component space, and you can see that generally early development was associated with a downregulation of corticosteroid genes. Later development, the up arrows were associated with a higher regulation of corticosteroid development sets overall. And then from the overlap between human and mouse development, we took the union here, and there were 118 developmental corticosteroid genes that were further evaluated for their association with asthma susceptibility. And here's the association. Here, basically in asthmatic subjects, you see an odds ratio of about 2 for corticosteroid developmental genes versus not. So significant enrichment for asthmatics. And here are the 8 genes that were noted in all 3 of the data sets in both mice and in humans. And you can see that from there, 3 of the 8 were significantly associated with asthma association, and a couple more were also associated with asthma treatment response in bronchial biopsies. Okay. So, in summary, the genes that are important in lung development are significantly enriched in both vitamin D and corticosteroid response genes. Most of the vitamin D genes are upregulated across gestational times. Corticosteroid response genes are equally up and down regulated over time. And a significant portion of both the vitamin D and corticosteroid genes are differentially regulated, during development, are also differentially regulated in relation to asthma susceptibility. So in conclusion, certainly the study of genomics of lung development during the period of branching morphogenesis, I think it does hold some real promise for identification of novel molecular determinants for both lung function and airways disease, specifically asthma. And using principal components, we have identified both that the vitamin D and corticosteroid pathways are clearly overrepresented during development, very important modulators of development, and are associated with asthma and their related outcomes. And these are the folks that have been working primarily on this. I think I'd like to specifically acknowledge Alvin and Sunita, who've been doing yeoman's work in terms of most of the analyses related to this. And I think we're going to wait until the end to take any questions.
So I'd like to thank Dr. Fanta and the Partners Asthma Center for giving me the opportunity to talk about our work looking at the effective in utero smoke exposure on this developmental timeline. As Dr. Tantisira just gave you a broad overview of how the transcriptomic profile changes over the time course of lung development. Now what I'm going to do over the next 10 minutes or so is give you just a broad overview of how in utero smoke exposure, which is a known intrauterine environmental perturbation, affects the genomic signature of lung development, how this genomic signature persists into the postnatal period, and its association with asthma susceptibility. It's no surprise to anybody in this room that cigarette smoking remains the leading cause of preventable deaths in the United States. In spite of all of the public health measures that have been aimed at tobacco cessation, an estimated 13.8% of women continue to smoke during pregnancy, with state-specific rates as high as 35 to 40%. Of concern, in utero smoke exposure remains among the most common preventable causes of infant morbidity and mortality. And there's a preponderance of epidemiologic data showing that in utero smoke exposure is associated with early onset wheeze, lower lung function, increased airways responsiveness, and asthma susceptibility. In spite of this overwhelming amount of epidemiologic data demonstrating this link, the biologic mechanisms underlying these associations are really not well described. Based on these epidemiologic observations, as well as our knowledge of the transcriptomic profile of normal lung development, we hypothesized that the gene expression profile of in utero smoke exposure, or IUS, during early human lung development is associated with asthma susceptibility later in life. And over the next several minutes, what I'd like to do is just give you a broad overview of the approach we're taking to address this hypothesis. In part one, we really look at just differential expression analysis based on in utero smoke exposure in the human fetal lung tissue samples that Dr. Tantisira previously described. In part two, we take a network-based approach. So we know the vast majority of complex diseases are really not related to a single gene or single perturbation in a single gene. So network-based approaches give us the ability to look at gene-gene interactions and how these interactions change in the face of environmental perturbations to identify groups of genes that are influential in terms of disease susceptibility. And in part three, we take a look at this in utero smoke exposure module and look for its persistence into the postnatal period and look to see if it's associated with asthma susceptibility. And I'm going to go into a little bit more detail on all of these fronts. So using genome-wide gene expression profiles available in our human fetal lung tissue samples as described by Dr. Tantisira in the last talk, we are able to infer an individual's in utero smoke exposure status based on a nicotine metabolite. So we're able to do this based on the fact that nicotine enters the fetal circulation via the placenta, but it really is not known. It's not known to be metabolized by the placental tissues overall. So we can use placental cotinine values, cotinine, a well-recognized nicotine metabolite. So we can measure nicotine or the placental cotinine value as a marker or biomarker of in utero smoke exposure. So in collaboration with our colleagues at Children's Mercy Hospital in Kansas City, we measured the placental cotinine values for each of the placentas of the concurrent fetal lung tissues and identified a cut point of 7.5 nanograms per gram tissue, which gives us a sensitivity of 78.7% and specificity of 100% for identifying these fetal tissues as in utero smoke exposed or non-smoke exposed. Of note, this cut point also identifies a cut point at which xenobiotic metabolizing enzymes in the fetal lung tissue are also upregulated. So in the analyses that I'm going to describe from here on forward, we use a subset of our human fetal lung tissue samples that are age and gender matched to try and decrease any of the other coexisting noise in these samples. And we identify 53 that are non-smoke exposed based on a placental cotinine value of 0 nanograms per gram tissue, and 51 that are in utero smoke exposed based on the placental cotinine distribution that you see here. We performed differential gene expression analysis based on IUS exposure using linear models adjusted for post-conception age and gender. And we identified 154 genes that were differentially expressed by in utero smoke exposure. As you might expect, based on the sex-specific differences that were outlined in the previous talk, there were also some notable sex-specific differences in gene expression changes based on in utero smoke exposure. Notably, male subjects had an enrichment of genes that were differentially expressed by in utero smoke exposure. We were actually able to identify roughly 5,000 genes that were differentially expressed in the male samples alone. So pathway enrichment analysis and the genes that were differentially expressed by in utero smoke exposure demonstrate some upregulation of some interesting pathways, including pathways in cancer, including small cell lung cancer. So as I previously stated, the vast majority of complex diseases are not related to a derangement in a single gene alone, but are really the result of a complex interplay between multiple genes as well as environmental perturbations. And so network modeling approaches are now being used to model this complexity. And weighted gene co-expression network analysis, or WGCNA, is one of those modeling approaches. And very, very simplistically, WGCNA is a method for describing the correlation patterns across gene expression samples. And this method allows us to define modules or clusters of genes based on co-expression of these genes. We're able to determine the preservation of these modules across networks. And then we can relate these modules to external sample traits, including in utero smoke exposure. So we performed WGCNA in our human fetal lung tissue samples and identified 37 modules or sets of co-expressed genes in this analysis, a subset of which you can see here. So each of the modules is represented based on this algorithm is identified by a color. Of the 37 modules identified, 6 were associated with in utero smoke exposure. And I'm going to bring your attention to this module here, the midnight blue module, which is a module of 38 genes that is significantly associated with in utero smoke exposure at a p-value of 9 times 10 to the negative 4th. I'm going to talk about this module in more detail. So now we've identified the genomic signature of in utero smoke exposure. The question becomes, does this module or genomic signature persist into the postnatal period? And is this genomic signature influential in terms of disease susceptibility? So based on this data, we really hypothesize that the gene expression profile of in utero smoke exposure persists into the postnatal period and that it's associated with asthma susceptibility. In order to test this, we use a publicly available genomic repository called AsthmaBridge. AsthmaBridge is a multi-institution consortium that is geared at the genomic investigation of asthma. And as part of this biorepository, genome-wide gene expression profiles from whole blood are available in asthmatics and non-asthmatic controls. So we performed, again, differential expression analysis between individuals who had a history of in utero smoke exposures and those that did not, and identified that our fetal lung signature was enriched in this set of differentially expressed genes. Again, showing as kind of proof of concept that this fetal signature of in utero smoke exposure appears to persist in the postnatal period. We went on to look at WGCNA to see if that genomic signature of in utero smoke exposure identified by WGCNA, again, persisted into the postnatal period and was associated with with asthma susceptibility and AsthmaBridge. We did this by recreating the fetal lung WGCNA modules in the AsthmaBridge cohort. And what you can see here is that the midnight blue module that was identified to be associated with in utero smoke exposure in the fetal lung tissue samples is also associated with the history of in utero smoke exposure in the AsthmaBridge samples, and notably appears to be associated with asthma susceptibility as well. So we went on to look at this particular module in terms of its network content by visualizing it using Gene Mania. And one of the things that you can see here is that 2 of the key network regulators of this particular module is interleukin-2 and CD40 ligand, which you can see outlined here. We are in the process of doing additional work on these 2 network nodes in our mouse model of in utero smoke exposure, and Dr. Haley is going to go into that in more detail. So now we've shown you that the fetal lung signature, genomic signature of in utero smoke exposure identified during human lung development persists into the postnatal period, can be identified in the peripheral blood of subjects in the postnatal period, and is associated with asthma susceptibility. The question is, why do these genomic signatures persist? And one of the mechanisms that is postulated to be a result of in utero smoke exposure is changes in epigenetic modifications related to in utero smoke exposure. One of those potential mechanisms is DNA methylation. On a subset of our human fetal lung tissue samples, we have genome-wide methylation profiles. And in work that is being spearheaded by Dr. Al-Ghawari and Dr. Q from our lab, an association of this midnight blue module looking at the DNA methylation sites that influence the regulation of genes within this module demonstrate 13 significant associations between DNA methylation and the genes that were involved in this module. So this is kind of proof of concept that in utero smoke exposure has major influences on the genomic signature of lung development. Often, and at least a partial amount of these associations can be related to methylation and other epigenetic modifications. So we've been using this genomic investigation of in utero smoke exposure in human fetal lung development to identify genes that are modified by IUS exposure and are influential in terms of asthma susceptibility. And I think this type of work really shows that integrative genomic approaches in human fetal lung development can be used to identify novel susceptibility loci and really can help us disentangle the biologic underpinnings of the association between intrauterine exposures as well as disease susceptibility. And the future directions for this particular work is that we're validating, as I said, the IUS association with IL-2 and CD40 ligand in our mouse model of IUS exposure. A lot of people to thank for this work, including Alvin Koh, Divya Chabra, Amal Al-Ghawari, and Wei Lian Q, who have been instrumental in this work, as well as my mentors at the Channing. Thank you.
So I'm Kathleen Haley, and I want to thank Dr. Weiss and Dr. Fanta for including me in this presentation so that I can give you all just a little bit about what we do with the mouse models to amplify what the human studies that you've just heard about, and also to get at a little bit of mechanism. So with a nod to the local newspapers, I'm going to be going over the why, the what, and the how of mouse models and their utility in asthma. So why? Why would you study mice when you can study the human? What models do we use? And what have we learned from our mice? So first, the why, using animal models in any disease, is important because it finally lets you get at mechanisms. And this is particularly important for studying intrauterine smoke exposures and subsequent asthma propensity, because you need something that will let you to study developing lung throughout the whole range of development, and the mouse is uniquely geared or suited for those types of studies. Additionally, tobacco smoke and the effects of intrauterine tobacco smoke exposure or tobacco smoke byproduct exposure in the human has over 1,000 compounds that could be affecting different physiologic mechanisms. And the mice lets you look at the mechanisms, whereas the human study, no matter how elegant, really is more of an association study. Why look at mice for this? Well, their short generation times make them an ideal animal model for a laboratory. They have an extensive track record of laboratory studies, including use in tobacco smoke exposure studies that allow you to select the strain and also the time point that you would like to study. And because of the multiple strains, there's also the option of numerous knockout genes, transgenic mice. And so really, the mouse is an extremely good model. Are there any disadvantages? Well, of course. What might they be? Well, mice are not small humans, and the lung in the mouse differs a lot from the mouse and the human, including something as simple as the number of airway generations. Interestingly, the mouse is born at a very different stage of lung development than the human is, such that most of alveolarization in the mouse occurs postnatally, whereas it starts a little prenatally in the human. And perhaps most importantly, although the mouse is an extremely attractive model, mice don't get asthma. There are laboratory animals that get asthma, including cats and guinea pigs and horses. However, the availability of other types of models make the mice much more attractive to use than these other animals. So we use mice. So how do we study this? Well, there are two models that we use. One is a direct smoke exposure, and this is our old-fashioned but functional direct smoke exposure model that we had first used when Dr. Shapiro was here. And so the cigarettes go into these chambers, the mice go into these chambers. These are pumps The smoke goes up the syringes and then into the face of the adult mice. In this model, only the adult mice are exposed. There is also the Teague, which is the whole body box. It's much more efficient than this model. As you can see, you're smoking about maybe 10 to 15 mice at a time. With the Teague, you can smoke easily up to about 60 to 90 mice at a time in individual cages. You can also very readily look at neonatal smoke exposure if that's desired. Of note, in our models, we really focused on just the intrauterine exposure, trying to really kind of separate that out from the postnatal smoke exposure of the neonatal mice and the juveniles. In the asthma, we use an adaptation of Dr. Kobzik's and Dr. Fejelov's low-dose albumin model to kind of tease out the propensity of asthma development in our smoke-exposed mice. And how do we measure it? Well, standard analyses, histology, molecular analyses including protein and mRNA analyses, and also some physiology. You can use a rodent ventilator, which you can get a lot of detailed measurements of lung compliance, airway resistance. However, it's a terminal experiment for the mouse. Well, you can use Buxco, which is a little second, and there is some debate as to precisely what physiology you are measuring. However, it has the advantage is that you can measure it over time in one mouse. And so each of these different kinds of physiologic assessments have their uses. So what have we learned? Well, between our group and groups in other laboratories studying mouse models of intrauterine smoke exposures, we've learned that there are very specific age and developmental stage effects of the maternal smoking on the lungs. Particularly, the prenatal and the neonatal lungs are not just small versions of adult lungs. There's very specific regulation, and things that are true in the adult are just not true in the fetal or the neonatal lung. Maternal smoking has pleiotropic effects on the offspring. Gene transcription you would expect, but we also find that major regulatory networks, including epigenetic modification and alterations in miRNA expression, are also affected by maternal smoking. We find that the gene transcription affects multiple processes that are critical to lung development and maturation and postnatal functioning, including growth and development pathways, injury response pathways, including such things as toxic metabolite pathways such as the aryl hydrolase receptor, cytochrome P450s, and also large injury response pathways such as the autophagy mediators. We also find changes in immune pathways and immune cell function, which I'll be able to present some data for you. And interestingly, we also find that in our mouse model, the effects of the prenatal insult persist to a significant period through the postnatal life, which really adds to the highlight that prenatal influences can really affect disease in the postnatal and adult offspring. Other animals have also shown that nicotine or cigarette smoke in the prenatal stage will cause disruption in lung architecture. These are several labs over many times, and not just in mice and rats, but also in rabbits, guinea pigs, rhesus monkeys, and there will also show increases in pathways related to allergic airway inflammation. What we've found have extended the work of other colleagues in other institutions. And again, similarly, we found looking at H&E staining of lung tissue from P14 mice, so well past the alveolarization stage. The non-smoke exposed mouse has a normal appearing lung, a very beautiful lace-like appearance to the alveoli. The mouse with the intrauterine smoke exposure has larger airways, fewer airways because they're larger, but if you notice, unlike human adult emphysema, the interstitium is not lacking. There's robust interstitium here. And so it's more that the alveoli never formed properly in the first place. When you measure the size of the airspace using MLI analysis, there is a statistical increase. So this fits in well, that showing that our model is a very robust model. When we analyzed it further, we found that part of the problem seems to be related to the vessel number and development, and also the fact that the elastin, which is obviously incredibly important in the lung, is decreased. If you notice the large airway here at P14 IUS mouse, and again, P14, the neonatal mice, has never received direct tobacco smoke. The elastin is very much minimized in this large airway. Growth and development pathways, I wanted to show you a little about retinoic acid. So you eat something containing vitamin A, retinaldehyde dehydrogenase converts that to all trans retinoic acid, where it affects the nuclear receptors, the RARs, retinoic acid receptors, are XRs, which bind then to a response element, which are upstream of retinoic acid response genes. There's a negative regulator, COOP-TF, the chicken ova upstream promoter transcription factor, can also influence this. And if the animation works, as you see on the day of birth, not so many differences between the mice without IUS and the mice with IUS in several retinoic acid pathway elements. But at postnatal day 5, and this is an important time point because this is right before the development of the most rapid alveolarization, the mice with the IUS have significant decreases in multiple retinoic acid receptors, as well as the gene regulating the synthesis of all trans retinoic acid. When we looked at the protein, similarly for the receptors RXR, RAR, RAR beta, and RAR gamma, we find significant decreases in the expression of the protein in the IUS-exposed mice. Similarly, with the vitamin D pathway, which is critical both for lung development, as Dr. Tantisira showed, and for general immune responses, and is also uses the nuclear receptors similar to the RA system. And is very important for asthma development in our IUS-exposed mice. Again, quite a trend toward decreased vitamin D receptor. This is a quantitative PCR analysis. The expression normalized to 18S on the y-axis, postnatal age here, which is significant at day 5. Immune responses. For this, we use an adaptation of Dr. Kobzik's and Dr. Fedulov's model. Their model originally used a BALP-C mouse. We adapted it to the C57 mouse. A full dose, positive control dose, uses 2 doses of ovalbumin and then an aerosolized ovalbumin challenge. The low dose, which kind of teases out the subtle differences between the smoke and the non-smoke mice, uses a single dose and then an aerosolized challenge. And then for blocking, we pre-treat with anti-CD25. And very similarly to what Drs. Kobzik and Fedulov found, is that we find that the smoke-exposed mice, similar to their mice after maternal asthma, has a very increased eosinophilia in both the percentage and total numbers of the bronchoalveolar lavage after the single-dose ova. And as you might expect, both groups get a larger response with 2 doses. But interestingly enough, after anti-CD25, the non-smoker mice increase, as you would expect, because CD25 is constitutively expressed on T regulatory cells, which downregulate inflammatory cells. But the smoker response after these anti-CD25 pretreatment actually shows a decrease in the eosinophilic response, which was very puzzling to us, and we wanted to follow that up. Because why would blocking CD25 change allergic responses? And just to reflect back to Dr. Sharma's data, CD25 is known to be the receptor for IL-2. Well, it's expressed on activated T cells, but the anti-CD25 does block. Now, we could have a concurrent response, and it could last beyond the sensitization. However, you would expect that if we just block T cell responses, that should decrease the allergic response. It is on T regulatory cells, as we talked, and that's a response that we see in the non-smokers. But it's also on this cell type, an invariant NKT cell, which is a subtype of NKT cells. And interestingly enough, using primary splenocyte cultures from our P14 mice with and without IUS, cytokine responses after stimulation of iNKT cells show a consistent increased release of cytokines, both murine interferon gamma and murine IL-2 in our smoke-exposed mice, suggesting that these mice are primed to overreact to stimuli of the iNKT cells, which could increase their propensity to develop asthma. At the same time, the CXCL16 ligand, which is associated with iNKT cell activity, is increased in the IUS mice compared to the non-smoker mice in both protein by histology and also significantly mRNA. So I hope I've convinced you that using animal models, particularly the mouse model, can really add a lot of data and a lot of mechanistic information and really complements the human data and the transcriptomic and the other omic analyses that Dr. Tantisira and Dr. Sharma have outlined for you. And similarly to Kelan and Sunita, I want to thank an awful lot of people, including the Channing Line collaborators and a lot of technicians who have helped, and of course, my colleagues in the pulmonary department. And I apologize if I've left anyone out. And you can't do anything without money in the lab, and so we've had good support. And I want to thank you very much for your attention.
Okay, so can I have the speakers come up to the table here, and we can start to take some questions. And while everybody's getting ready here, my co-PI, Dr. Kopsik, is in the audience. So Les, do you have an initial comment you'd like to make about any of this, or?
No, I think I can have an initial question if you allow it.
Sure.
I wanted to ask Sunita about her presentation and the current concern in public health of a transition to e-cigarettes as nicotine delivery. And do you have any thoughts about what your approach, or what do we know about that kind of tobacco exposure in pregnancy? What might it have to do with lung development?
Do I need to do anything to, hello? I'm technologically unsavvy here. That's a great question. So I'll just repeat the question very briefly. It was the concern over e-cigarettes, how it's influential in terms of when being used during pregnancy, how it may affect the genomic signature. And the honest answer is we don't know. Right, I think one of the concerns is that it's going to change the genomic profile in terms of lung development. How that happens and what the actual influence is yet, I don't know. And I think that's one of the things that we would like to study ultimately. And one of the ways we could potentially do that is using things like the mouse model, but I can't say that I can answer that based on our current data. I anticipate that it would be extremely influential. We just don't know how yet.
I will add to that, Les, that we are in the process of establishing a new collaboration through Rachel Tyndall and a Alaskan population where they don't have e-cigarette data, but they do have a lot of Alaskan women, for whatever reason, like to do chaw tobacco. And so we will be able to potentially compare chewing tobacco versus cigarette smoke and potentially gain some insights from that perspective, at least.
So I have a question for Sunita. You identified a genomic signature in the cigarette-exposed fetal lung samples that's potentially predictive of lung cancer. Any possibility of a predictive test that you could screen smoke-exposed fetuses for the upregulation of pathway genes in those pathways and predict who might go on to develop lung cancer later in life?
Yeah, I mean, I think that's ultimately where we hope to take this investigation. I will say right now we have not done that work yet, but one of the potential ways we're going to look at this is looking at the signature. One of the things that I think is particularly interesting is that this in utero smoke exposure signature appears to be preserved in the whole blood of the postnatal individual. So we'll be able to actually attest this in some publicly available data sets in terms of different lung cancer data sets that are available on GEO. So maybe be able to create a predictive test, and that's ultimately the goal of this.
Okay, I have another question for Kelan. I outlined several other exposures that are potential epigenetic modifiers of the human transcriptome in terms of development. What are the challenges that you see going forward for this research with regard to being able to look at the interaction of these different exposures or measuring these exposures, and what are the possibilities there?
Well, I think the biggest challenge that we face in general regarding exposure is exactly that. If you're using aborted fetal samples for much of your population, how well are whatever levels that you're interested in, specifically reflected in the placenta or specifically reflected in other fetal organs. We were lucky in terms of the smoke exposure because we were able to actually get specific data on moms as a portion of the aborted fetuses. So we actually had some moms where we knew to what degree they were smoking, and we were able to specifically quantitate. I think that from that perspective, we are developing active collaborations with other folks. Cindy McAvoy has a prospective cohort looking at vitamin D levels, for instance, and vitamin C levels, for instance, and the role of vitamin C in effect modification developmentally going forward. So I think we'll need to take advantage of other folks that can help us along with those specific exposure outcome measurements.
For those of you in the audience who are not familiar with this paper, Cindy McAvoy, who's a pediatrician at the University of Oregon Health Sciences Center, published a paper in JAMA in May of this year, where based on rhesus monkey data, she treated pregnant women with who smoked with vitamin C and showed statistically that she could abrogate the lung function changes related to maternal cigarette smoke exposure by pretreatment during pregnancy with vitamin C. So it's really proof of concept for what we're trying to do with VDART with vitamin D in a very special population. And it does speak to this issue of the potential for modification of risk based on modifying the fetal programming. Kathy, some comments about, is there any potential, where do you think, where would you like to go next with the CD40 ligand IL-2 story? What are the things that we need to do there to sort of finish this off, and what other things would you like to explore in the mouse model related to the immunology?
Well, I think, I think that completing the IL-2 and the CD40 ligand story will involve, sort of facts analyses and sort of just taking the routine bench studies, looking at the mice to see if we can replicate and also Sunita's findings, and then also see if by manipulating the amount or using a knockout mouse, can we prevent the smoke exposure effects on the asthma propensity. I think that those would both be very good follow-up studies for that. One of the things that I think that would be very interesting, just to follow up on Sunita's first question with the e-cigarette, is that we do have a nicotine model, and nicotine has been used quite extensively to model maternal smoking. And it's quite concerning in view of the e-cigarette because in many ways, although it doesn't entirely replicate the effects of maternal smoking, many of the effects on the lung and some effects on the immune system can be seen after maternal nicotine. And so the e-cigarette concerns, I think, are quite valid.
Can I ask,
Sure,
Kathy showed us that the newborn mice from maternal mice that had been smoke exposed had different lung morphology. And I'm wondering, do we know in humans whether the same is true, or would you speculate that at birth the newborn from a mother who had smoked cigarettes has anatomic differences than
I don't know about the panel, but I can speak to that. There is a very interesting paper that appeared in the Blue Journal probably 10 years ago, where a group of SIDS infants, of smoking mothers, had their lungs examined pathologically, and the babies of those SIDS infants had subepithelial fibrosis that mimics the subepithelial fibrosis you see in asthma patients. And one of the questions that has come up is whether this is related to changes in physical forces that the fetus may have experienced during pregnancy, or whether it's some other mechanism. But the changes in the alveoli and the airways, other elastic recoil changes in the lung have also been described in human lung samples as well as mice. So there is evidence that there are structural changes, and that's what's so remarkable about this vitamin C story, that you could potentially reverse those at a physiologic level. We don't know whether they're really reversed at an anatomic level. Yes?
Question for Sunita. Were any of the exposed based on the nicotine biomarker in utero exposures resulting from environmental tobacco smoke as opposed to maternal smoking? And what do you think the implications of this are for that exposure?
Yeah, that's one of the things that we're actually very interested in looking at because of the two hit hypothesis, right? That you could potentially have intrauterine smoke exposure and then concurrent environmental tobacco smoke exposure in the early postnatal period that kind of works synergistically in terms of causing these abnormalities. And we do have a subset of individuals where we know that they had no, where the mom didn't have a smoking history, but say the father or another individual in the family was smoking. So we have an environmental tobacco smoke kind of covariate, for example, for a subset of these samples, so we can actually start to look at that. We haven't done it yet, but there is a small subset. And we're going to use those to look to see if we can see a dose-dependent phenomenon with the cotinine value, because I think just using the cotinine value alone, we can't actually determine if it was true in utero smoke exposure or environmental tobacco smoke exposure, but we hope to look at it kind of the dose-dependent phenomenon.
Yes?
Is there any critical developmental window that means the embryo is structured?
Yeah, so that's a great question. The question was, is there a critical developmental window? And one of the things that you could see from the data that Kelan showed is that there are clearly molecular inflection points that are different from the anatomic inflection points in terms of fetal lung development. And we have not, at least in the human, have not been able to define those completely because we don't have, for ethical reasons, fetal samples that extend throughout the full stage of fetal lung developments. But absolutely, there are so many things that are happening, and they're happening so fast, and this is going to be a major issue in terms of interpreting clinical trial data in this area, because when the trial started, in terms of fetal development, in terms of the intervention, is going to have a big impact on what's going on. Kelan's data that he presented, particularly the vitamin D data, most of the vitamin D literature would suggest that the effect on the fetal lung was in the third trimester, and his data clearly shows that vitamin D genes are acting at the stage of branching morphogenesis, and hence they're very involved in airway development. So, and that's obviously pseudoglandular canalicular phase as opposed to alveolar phase. So it's this issue of a developmental stage and when this is happening, and whatever the intervention is and whatever the levels are is also critical. The other area that's critical, I'm surprised that nobody's mentioned this, is potential interactions between these exposures. Sunita mentioned, or Kathy mentioned this, but vitamin D and retinoic acid compete for the RXR receptor. And although if you go into PubMed and you look at the literature, there's many, many, many articles about retinoic acid, and it's important for development, not just in the lung, but for many other organs. But there's only one article in the literature that documents that the effects on development of retinoic acid are different if there's vitamin D present than if it's absent. And vitamin D has independent effects. So the interaction between vitamin D and retinoic acid is critical. And vitamin D also is probably interacting with the omega-3 fatty acids. So there's a lot of interactions of these methyl donors during pregnancy, and stage of development is important. The levels of these donors throughout pregnancy or even at conception are potentially important. A lot of things that haven't been studied here that are potentially critical to having the most healthy lung you can possibly have when you enter the world.
I will add to just your initial question about specific, are there critical phases? Clearly, I think we all believe that early development is a critical phase. And what we don't know about later development, I think we're going to start finding out. There's an NHLBI consortium called the Lung Map Consortium, which was recently funded this past year. And one of their goals is to try and get some of the later stages of as close to normal lungs as possible and try and profile them using a variety of different mechanisms through early childhood to try and get an overall normal picture. Unfortunately, the one shortcoming for Lung Map is it sounds like they're really not going to have a lot of exposure data. And so it will give us a nice idea as to what should be happening. It won't give us really any idea about perturbations to the system.
The other thing that Lung Map is doing, which is critical, is that we've taken the approach here of summing transcriptome over all the different cell types in the lung. And what Lung Map is doing is cell-specific RNA-seq to try to look at activity within specific cellular domains, and that's obviously another area that's critical. Factoring stem cells, lung stem cells, into what is going on here is another area where this research could go that would be potentially important.
And I was just going to add that in addition to these comments, the other thing that appears to be of note, like if given some of our preliminary data that we've shown here and some other data that we didn't show today, that these critical determinants in terms of the molecular windows are likely to be different between the sexes based on some of the sex-specific differences that we see. So it's quite complex.
Yes, Kelan.
Has the bacterial fibrosis you mentioned that found in the cyst implant recapitulated in mouse model? Kathy working?
Say it again.
Is the bacterial fibrosis found in this implant has been recapitulated in mouse model?
Oh, the subepithelial fibrosis that I described in those human samples, has that been recapitulated in the mouse? I don't know the answer to that.
No, it has been, and it's also been recapitulated in other animal models of maternal nicotine, including the rhesus monkey model. And so it really seems to be a very consistent finding in the offspring of mothers who have nicotine or cigarette exposure.
Yeah, the reason I'm asking is Scott just mentioned about the stem cells. So epithelial cells contribute definitely for the subepithelial fibrosis, but the stem cell population is different from like mouse and human. So maybe that different population of stem cells could contribute differentially in mouse and human lung regeneration. So I think that could be possible to also consider to look at later.
Yes, Judy.
I just had a question about the vitamin D. Has it been assessed in populations, different ethnic groups in different environments? Because when I was in Minnesota, we found that many women, including women who were within childbearing age, had low normal vitamin D levels. Yet the population, there wasn't a higher significant prevalence of asthma compared to some African American populations. So I don't know, is it something that's been seen to apply to multiple populations?
So I would say two things. One is that the epidemiology is that about 67% of all people are vitamin D deficient. If we were to do the vitamin D levels of people in this room, 2/3 of the people would be deficient according to the old IOM or the Endocrine Society levels of deficiency. But the greater degree of vitamin D deficiency is seen in people who have high melanin content in their skin. So it's much more likely if you're an African American to have lower vitamin D levels than if you're Caucasian. That has been, the racial differences have been blunted somewhat by the fact that so many people spend so much time indoors and don't take supplements that levels tend to be low throughout. But in general, in epidemiologic studies, the populations that have the greatest health disparities have the lowest vitamin D levels.
That's a good place to end. Thank you very much.
Thank you, Chris.